So this concludes the third module. I want to do a quick review of the module. We started actually from germline DNA sequence. If you just sequence people's germline DNA, based on that, could you use epigenetics to help us understand, for example, which of the SNPs are important? Can you figure out the tissue origin of the disease and what target genes these SNPs regulate? And also based on all the SNPs, figuring out whether the patient is whether the individual is more likely or less likely to be sensitive to or susceptible to a disease, right? So that's kind of on the germline disease side. In terms of in reality, whether, for example, if we have a newborn, do we want to do their DNA test? I would say if the parents are not too worried about familial disease, you probably don't need to do this. The rare mutations that are going to affect the individual with a very strong phenotype, if the baby is born, it's too late. If they carry that, the mutation is going to happen. If they don't have it, most of the GWAS related SNPs are actually only giving you a very small percentage of increase in the disease risk. So unless you care really about familial related diseases, I think it's a serious decision whether the parents want to have the child tested. So potentially one side effect of testing individuals is that sometimes even if I don't test myself, if enough of my relatives are already tested, people can infer my genome already and assign me a probability of having some disease, and potentially insurance companies might give me a higher premium in order to get insured because they estimated from my relative that I might have higher risk of some diseases, right? And then most of this third module is related to cancer genomics. And so Professor Gettigas gave the lecture on mutation analysis, right? We do either whole exome sequencing or whole genome sequencing. You can use algorithms such as MuTech to make the calls. And at the end, you can have different format of the files. In the homework 6, you will be dealing with some of these mutation calls. We do not ask you to start from just FASTQ file or BAM file to get the mutation calls, because these are very computationally intensive tools, and they are usually done by big consortium. Even if you were to sequence your own DNA at a company or as a CRO, they usually give you the mutation costs directly, and they usually come in MAF or VCF files. The question is, how do you understand your mutation? How do you, in TCGA, how do you find the recurrent driver mutations and understand what that driver mutation might be doing, right? Then we talk about gain of function mutations for oncogenes, which make the cancer or oncogene constitutively active, or loss of function tumor suppressor genes, which stop preventing the tumor from uncontrollable growth. And you can use gene expression on top of mutation patterns to help you decide whether a gene is an oncogene or a tumor suppressor gene. Of course, this could be complicated. Some genes can be oncogene in some cancers, but tumor suppressor genes in other cancers. We can also look at cancer type specific mutation load. We mentioned that those cancers that are facing outside, such as lung or skin, and they also have later onset in patients, they usually have more mutation load. In addition, the mutation distribution can be affected by gene expression levels and also replication timing, which basically is a reflection of chromatin. The open chromatin regions are usually replicated earlier, whereas the closed chromatin, DNA methylated silent regions are usually replicated later, and those can accumulate more. The late replication, closed chromatin regions can have more mutations, but they are not necessarily driver mutations. They are just non-functional mutations. Another important analysis is copy number variation. So we mentioned GISTIC, which is a way to calculate per gene how often this gene is amplified and how much this gene is amplified or deleted. which can also give us information of whether some gene is an oncogene or tumor suppressor gene. And then we talk about subtype analysis. This mostly are currently only done cancer type by cancer type. Say within breast cancer, what are the subtypes? Within lung cancer, what are the subtypes? And most of those you can see from gene expression. You can also use protein expression, DNA methylation, microRNA, and sometimes mutations. But most of the current subtyping are done using gene expression. We also mentioned that the big surprise in genome sequencing in terms of identifying novel driver mutations are mutations on epigenetic regulators, suggesting that epigenetics is a very important mechanism to maintain the system identity. The cell knows its identity. It should have at least a reasonable growth, but once those controls are out, the system loses its balance. You can imagine when the cell is differentiating, it's kind of going down the ski slope. I think about patient developing cancer as if they kind of now get stuck on a ditch in a ski resort, right? So then the cell is in a totally unexpected stage. Also, from TCGA, we notice sometimes in subtypes of cancers, very unusual DNA methylation patterns, especially the promoter, it's a CpG island methylation phenotype. And very often these are related to specific mutations, viral status, or metabolic differences. And they can be associated with it, depending on the type of the SNP. They can be associated with better survival in some cancer types, but worse survival in other cancer types. And when you have tumor profiles of many samples in the same cohort, you can also use something like single sample GSEA or co-expression network to really look at gene modules. And then once we have the subtypes of different cancers based on gene expression, the very important thing is to look at survival. Do patients in different subtypes have different outcomes? Should they be treated with different drugs? Right. So for survival, we talk about log rank test, which is usually just one binary decision: yes, no, it has HER2 or not HER2 mutations. Right? Whereas if you have multiple variables and also there are continuous values, you can also use Cox-PH regression. For example, you want to see whether a collection of genes, their expression is related to patient survival. But for that, you just have to be careful. How big is your list? So in homework 6, we ask students to test if your gene list is big. Very often some random genes will also give you some survival differences. But so having a good simulation to make sure that, say, if we select 100 genes to have a good biomarker, it actually works better than just randomly selected 100 genes. Okay, so and then we talk about targeted therapy and drug resistance. I apologize, this year we were not able to have this lecture. I'll try to make up this recording later. So targeted therapy is just mostly drugs that can target oncogenes. For example, if you have EGFR mutation, then the patients are more sensitive, or the tumors are more sensitive to EGFR inhibitor. If they have ALK translocation, they are more sensitive to ALK inhibitors. If they have BRAF mutation, they are more sensitive to BRAF inhibitors. And in the last 20, 30 years, precision medicine is really exciting in identifying such mutations that you can create these are actionable mutations that you can identify a targeted drug. And so because of this, most of the major hospitals, cancer patients diagnosed will have a choice. Either they can get this for free, or they can have insurance pay for it. And some of them want to pay their own money to do the tumor profiling, look at the mutations, and based on that mutation, identify the right drugs to treat the patient. And this is really, I think, what precision medicine, it epitomizes precision medicine. And so very often these drugs target pre-existing mutations, and it will kill probably 99% of the cells that carries the mutation. But because of the DNA instability in the genome, in the cancer genomes, very often the 1% cells that resist to the drug will over time evolve to have either they previously has a small mutation already, and which made the cell to be resistant to the first drug, or over time new resistance can develop, or at least they get selected to be more abundant in the remaining population. Then the patient will develop resistance to the drug. And so combination therapy is the case where you can imagine if each of the drug target a subset of the cancer cells, maybe if you use 2 drugs or 3 drugs, then eventually all the cells will be killed. There are also cases for synthetic lethality, which is targeting, say, if A and B genes, knocking out each or inhibiting each of the genes separately have no phenotype. But if you target 2 genes at the same time, that really kills the cancer cells. That's a very, very important new way to target cancer cells with combination therapy, really to target synthetic lethality. Hopefully that will also kill more cancer cells, then the cancer cell will not have a fast enough response to develop resistance. And in order to do that, we talk about chemical screens. Basically, you can use different drugs to profile many, many different cancer cell lines. So the Broad Institute has generated this 1,400 cancer cell lines and treated with many, many different compounds at different doses. So you can see which cancer cell line. So each cancer cell line has also been comprehensively profiled with whole exome sequencing, with RNA sequencing. So you know the mutation, you know the gene expression. And then if the cell is treated with a chemical, we could see which cancer cell lines will be more sensitive to a particular drug using this IC50 measure. So the lower the IC50, the more sensitive is the cell to that particular drug. And so that will kind of tell you, if you see all of the cancer cell lines being sensitive to a particular drug, you ask, well, not all, but the subset of the cancer cells. You can ask, what is unique about those cells that are sensitive to the drug? And you might find, these cell lines harbor the same mutation. And then for CRISPR screens, it's a genetic screen, it's not a chemical screen. So this basically, you knock out the genes, which is, because sometimes your chemical is only targeting 200 possible genes, and the remaining genes you just don't know. But CRISPR screen, you really systematically evaluate every gene in the genome to see which cell line are sensitive to the knockout of a particular gene. You'll find cancer cells specific essential genes, which can be novel drug target. And you can also treat the cell with or without the drug and do the comparison for the CRISPR screens to see what are the biomarkers of response, what are the resistance mechanisms, and what are the combination therapies that can overcome the resistance. And with that, we also have this magic algorithm to analyze the CRISPR screens. We also briefly mentioned the depth map, which is a big data resource and also a very, very useful web server where you can look at all the over 1,000 cancer cell lines in their chemical screens and genetic screens. So on the course website, we have a tutorial, which is the depth map tutorial. I really recommend you to take a look at that tutorial, which can tell you how to really utilize this really a very valuable resource. And then we talk about cancer immunology, right? And also immunotherapy. So in order to really understand this well, we're looking at what are the mutations in each tumor. And then what are the mutations that are being presented on the cell surface? And in order to understand that, we need to use some tools such as either polysolver or OptiType to really call the HLA types for each individual samples. And then if we know what are the mutations, what are the HLA type, then we can use algorithms such as NetMHC to decide which of those mutations are really being presented by the patient HLA to the cell surface that then can be examined by the T cells and B cells. And we talk about immune cell infiltration. How do you estimate based on the bulk tumor expression, the level of different immune cell types in the tumor? We we mentioned this timer, which is initially an algorithm, but now it's a web server. We have applied 6 different immune deconvolution algorithms into one to make this web server. You can look at the estimation from each of the tools on TCGA tumor. You can also use the 6 different deconvolution methods and use your own tumor samples to make estimations using those 6 methods. And then also we talk about immune repertoires, which is the T-cell receptor and B-cell receptor repertoires. This can be in the blood, it can be in the tumor, and you can directly get it from commercial sources, but anytime you have a tissue RNA-seq, you can run the trust algorithm to get the TCR and BCR sequences for free. Even though we don't really get all of the TCR and BCR, we'll get the abundant ones, which are more likely to be the clonal expanded ones that are more relevant to recognizing the tumor antigens. We also talk about immune cell activation in order to understand whether the different cells are activated, what genes are differentially expressed. Nowadays, people not only do bulk RNA-seq, there are more and more single cell tumor RNA-seq data available. By this time, I think there are probably 30 to 40 tumor single-cell RNA-seq papers published, but I suspect there are many labs with similar data in preparation for a publication. So next year, we'll have a lot more. And also, there are new technologies such as single-cell attack-seq to profile tumor samples or other tissues to help us understand how the genes are regulated in those different immune cell types. And then we talk about how to predict immunotherapy response. What are the factors that influence how a patient would respond to immunotherapy? So mutation loads seem to be important because cancer cells with more mutations will look more foreign to the immune cell types. Having CD8 T cell infiltrating the tumor, that's very important. TCR diversity, especially in the blood, is important. It means at least you have some T cell that have the capability of recognizing different mutations. PD-L1 level in the tumor indicates that the T cells have already tried to attack the cancer cells. So the cancer cells have experienced some immune pressure. Then it will overexpress PD-L1 to stave off the immune attack. And so that's at least an indication that immune system has tried to attack. Interferon level is similar to PD-L1. It really is indication that the immune system have released some cytokines to kill the cancer cells. We also have an algorithm to measure how dysfunctional the T cells are. Initially, the T cells might be trying to attack, but it can get very dysfunctional. We consider this as like a dead drunk soldier. And so immunotherapy may not, or at least the current immunotherapy, may not be able to revive the dysfunctional T cells. And also T cell exclusion, which is many other suppressive cell types, such as macrophage, fibroblasts, and MDSC, which can prevent other T cells or other good immune cells from infiltrating the tumor. And so these are all very important. And so that's kind of a summary of that third module. In the third module, we still kind of describe some new methods, but also you can see right now in cancer genomics, especially cancer immunology, this is the hottest area where all the new genomic technologies really converge on high throughput sequencing, single cell analysis, right? CRISPR screens, and single cell sequencing plus CRISPR screens are really all being used in here, and computational analysis together with genomics. I think it's a kind of a very exciting field because not only the biology is fascinating, but also the technology is great and all the computational methods are really making a difference in this field. And that's where we are working on actively, because you can really see this being applied by many people and benefit their research.